suis ravie de célébrer avec vous les 30 ans de LEPSE et de faire un petit hommage pour la carrière distinguée de mon collègue François Tardieu. Merci Pierre pour m'avoir invité. So I will have a little introduction to growth kinematics, and that will lead to an appreciation of the role of LEPSE in imaging technology. I'll run quickly through an ecophysiology of kelp forests, which are really suffering in the Anthropocene, and that will lead again to an appreciation of the work of François. So I like to introduce growth kinematics by talking about curvature in the seedling hook. This was my first effort at mathematical biology. My postdoc mentor, Ralph Erickson, had urged me to familiarize botanists with the mathematician's definition of curvature. Botanists like to define curvature as an angle between a tangent to an apical part of the stem and a tangent to the basal part. If you look at this lettuce seedling, you see that it's got zero angle of curvature, and yet clearly it's curved. So mathematicians talk about curvature at a point on a curve in space, and they talk about the spatial distribution of curvature. So to analyze the curvature pattern, I chose a parametric form of the equation of curvature. You have the xy coordinates along the curve, and then you differentiate them with respect to a parameter, and I chose arc length as my parameter. So to do this, I took a divider and I marked off hundreds of equally spaced points around the outside and then the inside of this photographic projection. I was lucky to have the newest technology, the first commercially available digitizer. You click on a little button and the crosshair records the XY coordinates. So I then clicked on all these hundreds of points and differentiated locally and smoothed, and voila. Oh, this seedling hook does have curvature. It's true, it's straight at the apex and below the apex, but at the hook summit it has maximum curvature. And this pattern is preserved through time. And we'd like to know, I mean, this is ecologically an important adaptation, this hook. It's thought to protect the important dividing cells of the apex from abrasion as the seedling moves through the soil. And then when it reaches light, it flips open. So how does this happen? We need to do a growth analysis. And so we mark the stem. Oh, there it goes. Okay, now let's keep an eye on these marks. Oh, they're moving, and they got to the summit, this guy. And now he's moving past the summit. And now, oh, he's on the basal straight part. Oh, my goodness. So this simple marking experiment, we realized that the hook, which is not changing in time, it's made of a parade of changing tissue elements, and each bit of tissue that's produced first curves and then straightens. So this is reminiscent of fluid structures, which have a similar property that they may not be changing in time, but they're made of water droplets which move through them and experience rapid and continuing change. So I realized that concepts and numerical methods from fluid dynamics would be powerful tools in analyzing plant development and response to the environment. And so for the next few years, I did studies including deposition rates of nutrient elements into growing parts of leaves and roots with Bob Sharp and Ted Shao. We examined the mechanism of osmotic adjustment. We looked at all kinds of source and sink relationships. And very slowly, people started noticing some of these papers around the world. In 1993, I had some correspondence with a stagiaire in France, Bruno Mulia, who invited me to be on a scientific organizing committee for the first international meeting in biomechanics, which was to be held in Montpellier in 1994. And afterwards, I got a letter from another young French scientist called François Tardieu, inviting me to visit his lab and speak to his group. He had just started a laboratory about plant ecophysiology under environmental stress. What a thrilling invitation. And of course, I was delighted to visit. And those interactions with young French scientists turned out eventually to be among the richest and the most inspiring of my career. So in my work, I do a lot of time-lapse photography of growing plants. And so from my point of view, one of Francois's most important contributions was his avid support of always developing the most advanced image analysis platforms possible, and both in terms of equipment and personnel on all size scales. And this was from a paper of his talking about imaging at the subcellular level, at the organ level, at the canopy level, and even remote sensing, because more and more in this Anthropocene we're needing global pictures to monitor the health and to help the restoration of stressed ecosystems. So now I'll race through another curved structure. This was work with Mimi Cole on rules for primary growth gradients and tissue morphology in producing ruffles. We looked at the giant bullkelp. Now, kelp forests were a major ecosystem off the coast of California, all the way from Southern California up through Alaska. And if you look online, you can find some wonderful movies of these forests. They're incredibly productive. Here, enormous fisheries, enormous fixation of carbon dioxide. Here you see a sea lion. There are many, many invertebrates, oysters, crabs, abalone, sea anemone, and you'll see now the starfish, which is an important player in this ecosystem, and a sea urchin, which is a kelp eater. Here you see the kelp forest from above. And turns out these systems are in trouble. That's an orca, the killer whale. And now you'll see a shot of Friday Harbor Laboratory, where a lot of research goes on. So this is not a parochial ecosystem. It's found around the globe in all kinds of cold to temperate waters. You find, just in the subtitle, enormous kelp forests. The bull kelp that we looked at takes different shapes under different submergences and current speeds. It has a holdfast, which grabs the substratum. It has a stipe about 10 meters long, typically, with a float, and then these fronds are what we looked at, the photosynthetic organs. And it turns out that the fronds occur in several forms. In still water, you find these wide, ruffled forms. Where the site is exposed and there's faster current, you find thinner, unruffled, flat forms. And Mimi Cole, in her early work, showed that these forms are quite adaptive, that if you have a ruffle and a wide frond in swift current, it'll break because of the drag of the current. On the other hand, in still water, the ruffles prevent streamlining and clumping, and they allow maximum photosynthetic surface to be exposed. And she also showed that this was not a genetic difference, that if you, by transplant experiments, she showed that the form was induced by the current. So we looked more carefully at the form of the ruffle, and we found it could take several different shapes. You could poke it and make bigger wavelengths or smaller wavelengths. If you hung it, it made a helicoid. It had a twisted shape with a certain handedness. You could push to change the handedness. So we thought that probably these ruffles were made by what mechanical engineers called elastic buckling. So the hypothesis was that the growth would be a little bit more on the edge of the frond than along the midrib. And for a thin ribbon, bending is energetically much easier than stretching. So the minimal energy of the surface would be a ruffled surface. There were requirements for the elastic model. They should have, the ruffled blade should have a higher ratio of width to thickness. This is a morphological criterion. And that ratio should have a particular relationship to the lateral gradient in transverse growth rate. So yes, we found that the ruffles do have a much higher ratio. They're both wider and thinner than the straps. And then we did growth analyses, and these curves will be very familiar to students in François' laboratory, except that the frond was 2 meters long, and we had to go out in a boat to mark it, and we marked it by poking holes in it. Then we left the kelp to grow in the field for 6 days, went out again in the boat, found our fluorescent tags, and we were able to measure the displacement of the marks. And here is the growth analysis that anyone who's worked with me has to learn. The velocity field is the basic data set, and then you have to be able to integrate velocity following the holes, following the marks to find a growth trajectory, which is a very useful measure. So I better skip this. This should be familiar to you. This is kind of like a maize leaf. Growth was localized at the base of the leaf, and an element initially between 5 and 6 centimeters from the base over a 3-week period of displacement through the growth zone would get to be about 3 or 4 centimeters. And yes, there was extra growth at the edge of the ruffled blade, about 10% more. So the generalization from this was that the tissue adaptation the tissue transduces the signal of the swift current, the drag, in the growth zone. But it takes 3 weeks then, and you have to take the Lagrangian view again and follow the tissue out with time. If you want to figure out the physiology of this adaptation, you've got to look in the growth zone, not where you see the ruffles later on. So this resulted in a couple of papers. We did this work early in our careers and late in our careers. And when we went to publish the last paper in 2020, I was horrified to learn that those kelp forests, which used to dominate the subtidal off the coast of California for 500 kilometers, they were gone. And there are a number of reasons for this. There was a marine heat wave. There was an El Nino, which raises the temperature and also depletes the nitrate. But the main thing was that there was a wasting disease of the starfish, which preys on the sea urchin. And so there was a population burst in these sea urchins, and they devoured all of the kelp. Historically, the predators were sea otters and these starfish. And now we have 3 different kinds of kelp ecosystems. We have some healthy forests. We have a new ecosystem called the sea urchin barren, where the sea urchins have grazed all the kelp, and they go into kind of a resting state, and they're the only organisms there. And then there are refugia, which can be different sizes. So it's very patchy. All over the world, they're finding on a scale of a couple of kilometers, we can have different kinds of kelp ecosystems now. There are pressures of all different kinds, commercial catches, kelp harvest for food, even for potash. There's an ag interaction because unmanaged grazing can lead to sedimentation. There are urban interactions, and so it's spatially varied. And whether or not you can restore these important ecosystems depends a lot on, oh dear, It's multi-scale, collaborative, many agencies. And now I want to talk about François. You Google to find out just how important he is, and you've come to an object lesson that you have to be careful when you use Google search. But really, you find that in his early work, he did lab and field work always together, always concerned with environmental physiology, very interested in development in terms of underlying processes. This paper with Alim Ben-Haj Salah was particularly interesting to me. Also, the more recent work of Olivier Turc, beautiful work on the silk of corn. And then a new technology multiplied the number of genotypes that could be tested. This has been very influential work headed up by Christine Granier. His models unify across spatial and temporal scales, and he has headed and been involved in collaborations more than on the local scale, agencies, countries, and continents. So thank you, François. You have been a pioneer in all these efforts. You have seen the coming of the Anthropocene, and you have exerted enormous foresight, hard work in both the field and the lab, a multidisciplinary approach. You reject all those academic silos, genetics, math, hydrology, soil science, agronomy, devotion to crop production and water protection, and you have the ability to move across scales from genes to plants to labs to fields to ecosystems, and you have been a brilliantly skillful administrator locally at LEPSE, at INRAE, and across countries and continents.